Imbibers, a very good afternoon. So welcome to today's session. So children, in the previous lectures we have discussed about the need of classification and how classification came into the limelight and how different classification was given by different scientists, and each and every classification was defined on the basis of respective criteria, but each and every kingdom of classification has their own drawbacks and has their own disadvantages. So the widely accepted kingdom of classification was given by R.H. Whittaker. He introduced the 5 kingdom classification, and he actually put the monerans, the whole prokaryotic unicellular organism, a very primitive organisms, into kingdom Monera. But eventually when the 6th kingdom classification came into limelight, what he did, he actually divided the kingdom Monera into other sub-kingdoms, what is the Archaebacteria and Eubacteria. So in the today's lecture only we are going to discuss about that only, that why Archaebacteria and Eubacteria were put into respective sub-kingdoms and not a part of typical kingdom Monera. So before we start, let me introduce you to myself. I am Anam Khan. I'll be your botany expert for the session 2023 and 2024. We are going to discuss all the amazing and interesting topics related to your botany part. Okay, so let's start our today's session. So in the today's session we are going to discuss about the Archaebacteria and the Eubacteria, the other 2 sub-kingdoms of the Monera. Moving forward, children, this whole classification constitute of everything. Of course, the 5 kingdom classification. We will be going through the kingdom Monera. After it gets over, we are going to discuss about the Protista. Then we will discuss about the fungi and kingdom Plantae, and of course kingdom Annelida will be discussed by your zoology expert. And we also have viruses, virions, prions and lichens to discuss. Okay, so we are going to discuss all these topics in the respective sessions. Okay, but in today's topic we are going to only discuss about the kingdom Monera and the other 2 sub-kingdoms, which is the Archaebacteria and Eubacteria. Talking about the Archaebacteria, let's see what are the journal feature it possesses which makes it different from the monerans or maybe the Eubacteria, why they was put into a sub-kingdom. So the journal features of an Archaebacteria is that basically the Archaebacteria are the ancient bacteria, or simply I can say the oldest living organisms on earth, owns only a single cell. Yes, the ancient bacteria. Yesterday also we have discussed about it, that the primitive organisms or the millions, life has been on earth like billions and millions of years ago. But the first existence on our planet, the first life on our planet was basically considered that it might be the bacteria. So yes, the Archaebacteria is the oldest living organism on earth, with having only single cell performing all the life activities. Okay, simply they are obligate as well as facultative anaerobes and prefer to grow in anaerobic conditions and can survive in the presence of oxygen. Okay, they can be obligate, they can be facultative anaerobes, but they can be according to the respective requirement. If they need, they might survive into a oxygen, in the presence of oxygen also, because right now the condition we are talking about our earth is that we have a lots of oxygen and all the living organisms are majorly surviving on the organisms. So Archaebacteria might show a shift, and they have shown a shift from typically facultative or obligate anaerobes to someone who takes respiration of basically and can grow in presence of oxygen also. Okay, they are also known as extremophiles, means extremely harsh condition, extremely typical, difficult, harsh, whatever you can say, whatever adjective you want to add to this, that they can survive in such conditions. So they are also known as extremophiles. Extremophiles, some organism which can survive in extremely harsh conditions. Those are also Archaebacteria. And yes, they belong to kingdom Monera and are classified as bacteria. They do belong to the kingdom Monera after the Carl Weiss was describing that it also majorly nothing, the whole kingdom Monera just has two subdivisions, which is the Archaebacteria and Eubacteria. So yes, it is a part of kingdom Monera only because it is finally after the Whittaker it was given a subdivision, but yet it still remains a part of a Monera. Okay. So these are the few general characteristics of Archaebacteria, that they are extremely, they can survive in extreme conditions, they can be typically obligate and facultative anaerobes, but they can also survive in the presence of oxygen, and they are the oldest living organisms on earth which owns a single cell. Okay. Moving forward, discussing about the general characteristics of Archaebacteria, next we have a question. So the Archaebacteria, what it is? All halophytes, all chemoheterotrophs, earliest known organisms or fossils? A very simple question which has a very simple answer, that all these Archaebacteria fall into the category of which? They have which category? All the Archaebacteria comes in which category? Come on, quickly answer. Asif, Dhanush, T, Princess. Children, this is so easy. Just you have to put, just now I have told you what is the answer. Okay, the Archaebacteria are typically the earliest known organism, means they are the most ancient. Archae means ancient bacteria. They have been surviving here for long back. They are extremely primitive and they are also known as the earliest known organisms. Okay, so moving forward, children, next we have another question. I think this is something we have already done in the previous lecture. So just related to that, before we move forward, which part of the cell contain N-acetyl glucose amine? Yes, when we were discussing about the structure of the monerans, when we were discussing about the features of the monerans, which part of their structure contain the N-acetyl glucosamine? Just being little bit sharpening your memory. Yesterday only we have discussed about it. So easy, absolutely easy. The N-acetyl glucosamine. N-acetyl glucosamine. The N-acetyl glucosamine is a part of the cell wall. Yesterday only we have discussed the cell wall is basically having both proteins and glycogens. Okay, phospholipids or peptidoglycans, sorry, not phospholipids, peptidoglycans. Okay, so they have both peptide amino acid, chain of amino acid, and of course they also have the carbohydrate. So N-acetyl glucosamine is a component of cell wall. Clear? Next we are going to discuss about three major Archaebacteria existing. Three major Archaebacteria known, which have their own features, own method of existing and own habitat to basically live in. So the three major Archaebacteria existing is the Halophiles, Methanogens and Thermoacidophiles. Halophiles, Methanogens and Thermoacidophiles. Let's discuss each and every feature of these three major types. So the very first we are going to talk about is the Halophiles. So what are Halophiles? As the name suggests, I love salt. Halo means presence in extremely saline condition. Children, they are present in extremely saline concentration. Basically, the whole condition and the concentration should be rich in salt. So, talking about the Halophiles, they live in very salty environment and they occur in salt-rich substances like salt pans, salt beds, salt marshes, and they are typically facultative anaerobes. Okay, typically facultative anaerobes. So they are found in the high salt-rich conditions. They are typically present in the salt-rich conditions, which are salt beds, salt marshes, salt pans, and they are typically facultative anaerobes. Anaerobes means in the surviving in the absence of oxygen. They are typically facultative anaerobes. So what it is? This is I love salt. This is a bacteria which completely loves salt. Okay, it can only survive in the salty condition, extreme saline nature. Okay, talking more about the halophiles, children. The halophiles, how they are able to live in such extreme condition. What is the reason? Can you imagine some organism which is living in extreme saline condition? Simply, children, in general words, it is in hypertonic condition, and you know nothing can survive in a hypertonic condition. It will lead to the what it will lead? It will lead to the lysis of the cell. Plasmolysis will happen. The cell death, the protoplasm will experience the death. The cell will not be able to survive in extremely hypertonic conditions. But how these halophiles are able to survive in such extreme saline conditions? So the reason they are able to live in extreme high salt concentration, presence of special pigment called bacteriorhodopsin. Please remember this. There is a special pigment present in the halophiles which is called bacteriorhodopsin, which is present in the plasma membrane, which produces ATP for metabolic activities, not for CO2 fixation. So this pigment called bacteriorhodopsin, which is possessed by the halophiles, which is present in the plasma membrane, which is producing an ATP, which is basically helping in the metabolic activities and producing ATP, but not CO2 fixation. CO2 fixation anywhere which is not necessary. Okay, it makes stable in high salt conditions, high salt concentration. It makes stability in the high salt concentration. Next, how they are able to survive? Because they have mucilaginous covering. Yes, they have this mucilaginous covering, okay, around their body, which is also helping them to survive in this extreme saline conditions. Okay, next, I said the cell will rupture, okay, but there is no sap vacuole only. There is no sap vacuole. Absence of sap vacuole leads to their survival in this extreme conditions. And they have high internal salt concentration. They have internally also have high salt concentration. So basically this is their nature only. Their nature only to survive. If you try to put them into certain condition which is not saline, they might die, because this is their nature, this is their habitat. Okay, they have adapted it, they have adapted all their functions related to that. So they are surviving in this such extremely salty, extremely saline conditions. And the examples of halophiles is Halobacterium, Salinarium. Okay, these are the just an example of the halophile. So talking about the Archaebacteria, which is an ancient bacteria, we have discussed. Okay, the general features we have discussed. So there are three types of existings, which is the halophiles, Thermoacidophiles and Methanogens. Okay, and we have studied the features of halophiles and how these halophiles are able to survive in extremely salt condition. That is also we have discussed. Okay, so let's see what we have in this video. As I click on this video, as you can see, this is cell wall, plasma membrane, bacteriorhodopsin, ATP, metabolic activity. Since there is a covering, the plasma membrane has a pigment called bacteriorhodopsin, which does the metabolic activities which led to the creation of ATP, which produces ATP, and as soon as it produces ATP, there is a cell wall, there is a plasma membrane, and there is this light, which just for your understanding only, there is no light producing. Clear? So this is all about the halophiles, the features of halophiles, how they are able to survive in extremely salt condition, and what exactly the content present which led to their survival. Okay, moving on to our next, we have a question. Yes, this is always important to solve question after you have known about the content. So children, topic is of halophiles and which of the following are found in extremely saline condition. What do you think? Archaebacteria, Eubacteria, Cyanobacteria, Mycoplasma. What do you think? Just put your some common sense, they're very easy. which are found in extremely saline conditions? Archaebacteria, Eubacteria, Cyanobacteria or Mycoplasma? What do you think can survive in extremely saline condition? Of course, the answer is Archaebacteria. Halophiles are a part of Archaebacteria only. That is why they can survive in the extremely saline conditions, because they are ancient bacteria, not the those organisms that survive in extremely saline conditions are known as halophiles. Next, the next Archaebacteria comes into our limelight is the Methanogens. So, what are Methanogens? Simply, the Methanogens are strict anaerobes and they are present in the marshy areas. What are marshy areas? If I say in Hindi, daldal jaisa area, जिसमें ज़्यादा there is lot of water, there is lot of sand, the mixture of it is like, what you say, that is the daldal kind of area, that marshy areas is where the Methanogens are present. Okay. What they do, they convert the formic acid and carbon dioxide into methane with the help of hydrogen. So, Methanogens, what they do, the Methanogens kind of a bacteria, they are converting the formic acid and carbon dioxide into methane with the help of hydrogen, methane gas. They produce methane gas by the conversion of formic acid and carbon dioxide with the help of hydrogen. So, they are creating the methane gas. So, children, as the methane gas is produced as a byproduct of this metabolism, this is their general metabolism which is taking place inside their body. But in that, as a byproduct, as a waste product, whatever you can say, these Methanogens are producing the methane gas. So, for them, methane gas is a byproduct, but methane gas can be utilized by us also. Okay, many industrial application is there of the methane gas, you can use them. Okay, so children, when the methane gas is produced by the Methanogens, it is being utilized for them, their metabolic product, their byproduct, their waste product can be of use. Okay, so these are the Methanogens. Talking about the Methanogens, children, they also live as a symbionts. I know there's something always coming into your mind, why ma'am is not talking about the Methanogens living in the herbivores, in the typical herbivores. So, children, the herbivores, especially the Methanogens Archaebacteria, live in a symbiotic association. What is symbionts? Something which basically help in the both are benefited from each other. They have this collaboration, they have this thing to live together and to benefit each other. So, the Methanogens are the nothing but are the Archaebacteria, which is Methanobacterium, live as a symbiont in the rumen of herbivores that chew their cud. Okay. So, the herbivores have this Methanobacterium living in their rumen, which basically are chewing the cud, typically herbivores only. Okay. So, children, what they help? They helpful in the fermentation of the cellophane. That is why you know what is gobar gas. The gobar gas is typically methane only, no? That is because when we burn that cow dung, it releases gas which is called methane, and that is because of this Methanogens only, Methanobacterium only. Okay. So, they finally convert methane by the action of Methanogens, and methane is a combustible gas. We can use it for the combustion, heat, to generate heat. Yes, the cow dung produces the methane because it is fumable, it is combustible. Yes or no? So, that is because of the Methanogens present in the rumen of herbivores or the rumen of cattle. Clear? So, the Methanogens, which is an Archaebacteria living in the mutual association in the rumen of cattle and producing the methane, which is a combustible gas when the cow dung is released. So, this is about the Methanogens. Anything which is not clear? Any questions related to this? Anything which is not clear, any questions related to this? So we have discussed about what is Archaebacteria. So can you children now you know about Halophiles, now you know about Archaebacteria, now you know about Methanogens also. So now you are able to relate to it that why we are keeping this in a subdivision, because these kind of a characteristics are not a typical characteristics of a bacteria. They are typically ancient, they have their own way of survival. Okay, so as you can imagine, that is why the Carl Weiss has kept the Archaebacteria into a sub-kingdom of kingdom Monera. Though they are bacteria only, but they yet show a different kind of what? Different kind of a behavior, different kind of an existence, different kind of existence in a habitat. They have their particular habitat to survive. So this is about Halophiles, Methanogens, and of course we have discussed about the Archaebacteria. Next come the question. So children, Methanogens belong to which part? Which part does the Methanogens belong to? Simple. Abhi right now we have discussed only the Methanogens belong to Archaebacteria, because Methanogens is a part of an ancient bacteria. Okay. Moving forward without wasting any time, talking about the third Archaebacteria, which is the Thermoacidophiles. So children, I know the name is very much clear. These Thermoacidophiles, as you can see, yes, this is where they are surviving only, extremely hot condition. Okay, so children, Thermoacidophiles are the organisms or bacteria which can grow at extremely high temperature up to 18 degrees C and the pH as low as 2. Very, very important. This point is very, very important. Please, very important. What are Thermoacidophiles? Thermoacidophiles, they can survive in the extremely high temperature which is of 80 degrees C and can survive up to 2 pH, which is extremely acidic condition. So they are Thermoacidophiles. Thermo means temperature, acido means acid, and phile means basically are living organisms. Okay, so Thermoacidophiles, thermal condition, acidic conditions, they are able to survive in such extreme condition. They are present in hot sulfur springs and geothermal vents. And the example of Thermoacidophiles are Thermoplasma, Sulfolobus and Thermoproteus, and common is Thermophiles. So children, these are the few features of the Thermophiles which is very much important for you to remember that these Archaebacteria and that these types of Archaebacteria are showing such different kind of a characteristics. So the Thermoacidophiles, they can survive up to 80 degree temperature. Imagine 40-42 mein to we are only just sweating very much. Okay, we can only not survive if the temperature rises to 42, 43, 44, 45. Okay, imagine they are surviving at 80 degrees C and they are present in the hot sulfur springs and the geothermal vents. Okay, so this is about the 3 types of Archaebacteria and the general characteristics of Archaebacteria. I just really hope that now you are able to make out why the Archaebacteria is not a typical component of Monera and are having their own subdivision of Archaebacteria, and the type of and the characteristics they possess basically are little bit more extreme. All 3 of them show the extreme characteristics. Okay. Okay, so this is about Archaebacteria and Eubacteria. Again the question, the primary producers of deep sea hydrothermal vents ecosystem are, what do you think? The primary producer of deep sea hydrothermal vents, what do you think are present in the deep hydrothermal vents? Yes, of course, the chemosynthetic bacteria, which is also our thermoacidophiles, they are present in the deep sea hydrothermal vents, not green algae, because green algae needs, they do the photosynthesis, not blue green algae, and the coral reefs, of course, not possible. Okay. Next question. The chemosynthetic bacteria obtain energy from, what do you think, these thermoacidophiles and these halophiles are obtaining their energy from? Sun, infrared, organic or inorganic chemicals. Chemosynthetic bacteria are obtaining energy from where? Children, the chemosynthetic bacteria are obtaining energy, very simple, very basic, from the inorganic chemicals. That they are obtaining their energy because they are chemosynthetic, that's why they obtain their energy from the inorganic materials. Okay. Next, you need to fill in the blanks. The correct option given in the bracket Archaebacteria is known as, Archaebacteria are known as living fossils, coelomic organism or heterotrophic organisms. Archaebacteria are known as, what do you think Archaebacteria are known as? So, Archaebacteria are typically known as living fossils because they have been surviving on this planet from millions years, billions of years ago, and that is why they are also known as ancient bacteria. They are also known as ancient bacteria. Coming to our the next topic, which is the Eubacteria, since we have already discussed about the Archaebacteria and the types of Archaebacteria. Now comes to the second, or comes to our subdivision, the another subdivision, which is the Eubacteria. What is Eubacteria? So children, it is also known as true bacteria. True bacteria means the first true bacteria which is extremely showing real bacterial characteristics. Okay, extremely original, true bacteria. Okay, so Eubacteria is also called as true bacteria. Why? The peptidoglycans can make up to a cell rigid cell wall. The peptidoglycan, you know that yesterday only we have discussed, the cell wall of a bacteria is made up of peptidoglycans. So, the cell wall of Eubacteria is also made up of peptidoglycan. Pili. What is pili? Pili in some bacterial cells a surface that aid them in sexual reproduction, because also known as sex pilus. Okay, F negative, F positive. These are sex pilus or pili basically present on the surface of the bacteria, which help them in the process of conjugation or parasexuality. Yesterday only I have told you the false sexual reproduction, okay, which is the parasexuality, is because of this pili present on the surface of the bacteria. Okay, a typical pathogen can adhere to a host with the help of pili cell. Of course, they can stick to the host body because of this pili. They are very sticky, they just hold on to the host body, they can stick to it. And the further Eubacteria are divided into two categories, gram positive and gram negative. Yesterday only we have discussed about this, what is gram negative and what is gram positive. So, now you see the typical characteristics of Eubacteria, that they are true bacteria, cell wall is of peptidoglycan, they show parasexuality, they have pili on their surfaces, and they are also known as gram positive and gram negative bacteria. So, these are the few features of Eubacteria, okay, and that is why it is known as true bacteria. Okay, I hope this is very clear, because now you know this is what the session was all about, that why Archaebacteria and Eubacteria are not a part of a typical monerans, why they are having their subdivisions. The monera is further subdivided into two, which is Archaebacteria and Eubacteria. I think I missed this. Can you see this? This is gram negative, children, this is gram positive. Can you see, in the gram positive, whole cell wall is basically stained, that is why it is known as gram positive. Okay, here complete stain, a very light stain they show. So gram positive and gram negative is different on the basis of their cell wall which is taking up the stain or not taking up the stain properly. So gram positive which takes up the stain, gram negative which does not take up a stain properly. Now the question comes up, in eubacteria, a cellular component that resembles a typical eukaryotic cell. What do you think a eubacteria has and a typical eukaryotic cell also possess? Nucleus, ribosome, plasma membrane or cell wall? What do you think is a common component of a eukaryotic cell and a eubacteria? What do you think is a common component of typical eukaryotic cell and eubacteria? Of course children, very simple, the plasma membrane. Plasma membrane is something which is very much common between the eubacteria and eukaryotic cell. Okay. Moving forward, next we are going to talk about the mycoplasma, the other type of little tiny structures present with their own characteristics. So children, talking about the mycoplasma which is there also in your NCERT. So let's know about little bit about the mycoplasma. These are extremely infectious. They are extremely infectious. They might be very tiny but they are very infectious. They have pleomorphic, means pleomorphic, different shapes. Can you see this, different shapes and sizes? This is what pleomorphic means, different morphology and they can change their shape. That is why they are known as pleomorphic. Okay. It is a bacterial genus that lacks a cell wall that protects the cell membrane. It means the pleomorphic is highly infectious because they do not have a cell wall. This is also a thing, because the pleomorphic, the mycoplasma is extremely infectious because they do not have a cell wall that protects the cell membrane, and they also replicate by binary fission. The cytoplasmic division results in the multinucleated filament development. What is cytoplasmic division? When there is a cytoplasm, when the division takes place, it creates a multinucleate condition. Multinucleate means more than one nucleus is present, which led to the filament development. Okay, filament basically whole rod like structure, that is what you call filament, which has a multinucleate condition, which is more than one nucleus is present in a stalk like structure. Okay. Due to lack of cell wall, they naturally resistance to drugs. This is what I said. Due to the absence of cell wall in the mycoplasma is extremely infectious in nature, because that is the reason they show resistance to drugs. Okay, drugs, whatever, life-saving drugs. Okay, the mycoplasma does not have a nucleus and other membrane-bound organelles. They do not have nucleus and other membrane-bound organelles are also not present. Okay, other membrane-bound organelles are also not present in the mycoplasma. So this is also more of an extension of the bacteria, showing little bit features but yet not a typical bacteria. So mycoplasma is also included in that. So mycoplasma has also have their individual features. Okay, little bit deviation from a typical bacteria. So the question comes up, why mycoplasma differ from bacteria in the absence of what bacterial cell has but mycoplasma doesn't have? Just now we have discussed what bacterial cell have but the mycoplasma doesn't have. What do you think a bacterial cell wall has but the mycoplasma doesn't have? Of course children, the cell wall, just now we have discussed, the cell wall is a characteristic feature of a bacteria made up of peptidoglycan. On the basis of that, gram negative and gram positive is also created, but yet the mycoplasma does not have a cell wall, and extremely. Now comes the blue-green algae, which is the cyanobacteria. Let's see the few features of cyanobacteria. So children, cyanobacteria, which are also known as BGA, blue-green algae is typically photosynthetic autotrophs, and they have chlorophyll pigment just like green plants. Cyanobacteria are completely autotrophic because they possess chlorophyll-like pigment that is present in the green plants, which is helping them in doing their photosynthesis, or basically having or obtaining their own nutrition or synthesis of their own nutrition. The plankton found in the oceans and seas are cyanobacteria. They produce a major fraction of the oxygen in the atmosphere, and that is why it is also known as first oxygenic bacteria. Cyanobacteria is also known as first oxygen producing oxygenic bacteria, because they were assumed, they were said that they were the first organisms on the earth who does the photosynthesis and produces oxygen as a byproduct, which led to the survival of all other aerobic organisms on our planet. So they are also known as oxygenic bacteria. They can reside alone or in colonies. As you can see, they can have a colony or they can live alone. Asexual reproduction through transverse or binary fission, and they have few specialized structures for locomotion, which helps in also in the reproduction, which is the actinids, hormogonia, homocyst and spores. But these are structures which are only present in the harsh condition. It means these are the few alternative form of reproduction through the actinid, homocyst, spores. They're only heterocysts. These are only producing method of asexual reproduction, especially in the harsh condition. Whenever there is harsh condition, it will lead to the production of, or it will help in the reproduction of cyanobacteria. These are the few specialized structure which led them to always reproduce also in the harsh and extremes condition, and that is the reason they have survived it for so long. Okay, so cyanobacteria is also known as first oxygenic bacteria because they are the reason also to produce lots and lots of oxygen on our planet, which led to the existence of aerobic organisms. Clear? The next comes the question, the unicellular cyanobacteria reproduce asexually through which process? Just now we have discussed. Asexual, they might be having some different method to reproduce, but they also show the reproduction through which? Of course children, the binary fission. Binary fission is a basic method of reproduction in bacteria. Okay. So the next question comes up, the next question doesn't come up, then we are end to our today's session. So children, now I hope you have understood by Archaebacteria and Eubacteria, what are the features of Archaebacteria, what are the components of the Eubacteria, what is Mycoplasma and what is Cyanobacteria. All these things are a part of your NCERT, okay, and we have discussed everything in very detail. Okay, tomorrow we are going to discuss about the Protista, we are going to discuss about the kingdom Protista, so do not miss that class, we will meet at the same time. So this is something which we have already discussed. The Archaebacteria, Halophiles, Methanogens, Thermoacidophile, Eubacteria, true bacteria, of course on the basis of gram positive and gram negative. Halophiles lives in salty condition, Methanogens in the typically marshy areas, Thermoacidophiles they are surviving in the extremely hot and low pH condition. So now you know the Archaebacteria and Eubacteria are the subdivisions of kingdom Monera, and these are the features they possess. These are the types of Archaebacteria and Eubacteria they have. Okay, and that is why they are put into subdivision by the 6 kingdom classification. Okay, so children, before we end this class, you know this is our Embibe app. So children, this is one-stop solution on everything, and you can change your board, respective board you can study according to whichever board you belong to. You can basically select everything. This is the place, as you basically click on this, you can have all the respective boards present. As you see children, these are all the boards present in the Embibe app, and whichever board you belong to, you can basically select, and all the classes are live already taking place on the Embibe app. Let me introduce to that. I have selected CBSE, and since you belong to 11th standard, I'll click on the 11th CBSE and I play next. I can choose my respective language. I'm comfortable with English, I'll choose English. I'm comfortable with Hindi, I'll choose Hindi. Okay, and as soon as I press done, I will land on this page. This is a home learn practice test, and as soon as I press on the home button, children, I'll land on this and you can see the respective buttons: my home, Embibe live classes, revision list and my timeline. As soon as I click on the Embibe live classes, all the respective classes which are going on, this is me right now teaching you. This is the next 7 days classes. Tomorrow we are going to study about the kingdom protista, so please register yourself for that class. And these all are the past recorded session which has already been taken. So if by any chance if you have missed any session, you can always visit our website, you can always visit our app, just download from the app store, and please just explore this whole one solution for your studies. Then you can also learn yourself respective videos, related topics and their concepts will be there, and you can practice different questions, you can create your own test, and you can always see how much progress you have made. Okay, and the next is our Embibe Lens app. So children, this is very unique, I would say. You just not have to only type, you just have to scan your textbook and all the keywords would highlight and all the related 3D objects will be visible on your Embibe Lens app. So please go download it, explore everything in 3D. Let's see, have a very visual learning because of this Embibe Lens app, and you can always join us on the app. Just download it from the app store. We'll meet tomorrow again at the same time. We'll start with the kingdom protista. Till then, please children, take care, keep imbibing. Bye bye.
Keep imbibing. We believe in you.